Okay, so welcome back, everyone. So if you're joining the session, that means you're interested in the PLINDER session. If you're here by mistake and you wanted to be in PLINDER for the time being, you would need to go back to the previous link. And most of you on the Discord channel hopefully already found the link to the Google Colab notebook, what I'm showing here. If you haven't, then I do invite you to go to Discord and navigate yourself to the Colab notebook. It will be much more interactive to follow it together as we execute. So first of all, what we need to do is to, once we start the notebook, we need to install the software. In my case, it has done that already. For you, doing it the first time might take a little bit more time. So we'll start from the top. And while you are installing the software, my colleague Tom can talk us through a little bit about the downloading of the dataset, how we would normally interact with it, and how we'll interact with it today. So Tom, when you're ready.
All right, thank you, Vladis. Are you going to drive, or would you like me to screen share?
I am happy to drive.
Okay. All right. So welcome, everybody. The PLINDER dataset is approximately 1.2 terabytes when it's fully downloaded and fully extracted. And so that, lends itself to some normal technical difficulties if you don't have that large of a hard drive. So there are a couple of features built into the PLINDER Python package that make interacting with the dataset somewhat more convenient, depending on your use case. The simplest possible thing that you could do is download the entire data set all in one shot. And to facilitate that, when you install the PLINDER package, we expose a command called PLINDER_download. It will take the current configuration and sort of execute the full download of the entire data set. There are three large portions of the data set where it will prompt you and ask for your confirmation of whether or not you want to actually do this download. And those are the systems archives, the protein similarity score data set, and the linked structures data sets. So those each represent some of the lion's share of the disk space required. And so you can, pass a -y flag to skip the confirmation, and it will assume that you want to download everything. Alternatively, you would have to, confirm the input prompt. So, we did try to wrap it in nice progress bars. They work to varying levels of success. So you have to be somewhat patient because it is a large data set if you plan to download everything all at once. So one of the features in PLINDER is that we can, instead of downloading everything all at once, lazily download only the files which you are requesting, in a given snippet of code. The side effect of that is we have to check whether or not that file has been downloaded and if it's the same as the one that's available in the cloud storage. That can end up being slightly cumbersome if you're trying to iterate over the data quickly, which is why we provide this PLINDER offline environment variable, which you can then set if you've already downloaded all of your data, and it will avoid that check that you're still in sync with the cloud. And so, that would be like a pro tip for advanced usage. Okay, so let's import some additional utilities that will be useful for visualizations in the notebook. And then we can talk about the so-called Plindex. So this is sort of the core structure that we need to slice and dice all of Plinder's useful annotations. However, it is rather large. So if you try to load the entire thing into memory, you'll need, sort of like workstation computational resources as it sits at about 24 gigabytes of RAM. So alternatively, we have this simple function called query index, which lets you sort of load into memory only the data that you need. And so as an example of how you would use the query index function, you can pass a list of columns. Now, the list of columns that you would be interested in are made available in both the hosted documentation as well as a sort of like a helper data frame, which we'll see further down, so that you can peruse the lists of columns that you may or may not be interested in, and then only load the ones that you want to inspect. This will, save you a whole lot of RAM, and you'll get much more performant response from the querying when you only load the data that you need. So one technical detail to note about the index is that system ID is not unique, because we have the possibility for multiple ligands within a system. There may be the possibility that there are multiple rows within the Plindex that contain the system ID. So just something to keep in mind. And yep, here are some summary statistics about the Plindex itself. 420,000 ligands, 310,000 systems that are unique, 108,000 bio units, and 77,000 unique PDB IDs. So that's super useful. In total, the index contains every possible Blender system that we could annotate. However, in the normal use cases, we're actually only interested in the systems which show up in the split. So recently we added the ability to sort of return only the contents from the Plindex that are contained within the split data set. So by default, only the systems that are in the train and veil splits are returned when you query the index. Alternatively, you could pass a splits argument to a query index, which would be a list. And the simplest argument could be a list of a string that is the star character, which would say, include all splits. Right. Oh, wow. Thank you. Yes, that's perfect. Okay, and so, moving on to more reasonable queries that we would want to make about the data, that's sort of the shape of it. And here are some, high-level important things to note about each individual system. So the, number of protein chains within a given system, most of them are single protein systems, but there are a good number of them that are multi-proteins. Similarly, for the number of ligands within a system, most of them are single ligand. However, that's not necessarily the case. And then we can, drill into the descriptions of the ligands, whether or not they're classified as ions or artifacts or fragments, as well as their cofactor designation. And so, yes, it's very much possible to, filter the collection of Plinder systems based on whatever you're interested in studying. And so maybe the simplest possible case to get started with is, single protein, single ligand systems. And so the way to get all of those, particularly for the training data, would be running, that cell block where we query the index for, only one ligand chain and only one protein chain. And then we see that we have, looks like 153,000 Plinder systems for which those filter criteria are true. And this maybe makes a good starting point for, the kind of data that you would want to try to feed into a downstream task of some sort. And then, so a number of ligand chains is one thing, but there are sort of like varying degrees of specificity for how you call a ligand. And so in PLINDER, we have this concept of a proper ligand, and we also have that annotated in the index. So you can alternatively query by the system proper num ligand chains, and that will return you the subset that are, meet the more stringent criteria for the classification of the ligand. Okay, and we see the record count there. Okay, and as I alluded to, there's the sort of the meta data frame for just inspecting the descriptions of all of the columns. So rather than having to load the entire Plindex to see what columns are available, you can just get all of the column descriptions, and then you can sort of, peruse this data frame to see, maybe search by keywords in the description column, or, filter by name starts with or something like that, so that you can then pick out specific columns that you would pass to the query index function itself. And it's an exhaustive list, and it contains basically all of the annotations to date. And so this would probably be your best starting point for, digging into the full availability of the annotations in the Plindex. So with that, maybe I will pass it back over to Vladis to dig deeper into the types of available annotations. And
yes, thank you, Tom. This is very helpful. So as Tom already mentioned, there's plenty of these annotations. There's more when we have time to go into deep explanations for and motivations for keeping it. However, it is extensively documented in our website, and we hope that in our documentation website, and we hope that you will visit that whenever you decide which annotations are best to use filtering your data set. So many of the important annotations actually regard the quality of the data set. So like the input for our models, as well as the ground truth for our evaluations, is experimental data sets. And these are of varying degree of quality, as you can see from our publication. There are some systems that have alternative conformations for the ligand, as well as can have some missing atoms around the binding site. So in order to choose which systems are to be used for test purposes, if you want to have a stringent evaluation of your performance, it would be important to know some of the quality details. And them are listed here, and they can be queried also. We try to kind of like to group them based on the name. So the validation part is actually using the X-ray validation records. In this case, it provides you the descriptions of this is kind of like the resolution, as well as kind of probability scores, as well as a lot of other important data information that allow you to choose which system is suitable. So we just show this as a couple of examples. You might also want to look deeply only in the pocket part. So we also analyzed only the part that regards the pocket residues. So these can be mapped to individual residues, and we picked the ones that are within proximity of a ligand. So while some part of the system might be unresolved due to flexibility, or else the binding site might be fully covered by the density, and therefore this can still be considered as a high quality structure for protein ligand interactions. So that's all to be considered in the context of what kind of task you want to model and what kind of like how stringent of a test you want to perform. Our quality criteria, which in order what we call system pass validation criteria, and that's what we use for our curation of a high quality test systems, is as follows. It's a high resolution of 3.5 or lower, so kind of like and this a decent entry R value, which is this is more of a crystallographer's metrics to determine how well the model is the actual crystal structure data is represented by the model. One of the important part that is worth mentioning, that we try to make sure that all of the residues and all of the atoms surrounding the ligand are resolved. So like the ligand itself that is resolved. Also, it doesn't have clashes, doesn't have crystal contacts. And the residues that are around the pocket are also fully resolved. So kind of like this is an important note. There's only a small exception for covalents, because sometimes covalents have a leaving group, so you will have some of their heavy atoms will be lower for those ligands, but we allow it as an exception for covalents. So we can look into this particular query. In this case, now we query for the splits that include train, val, and test, and all of those that we also interested how many of them pass the validation criteria that is currently defined above. So we can actually plot, and we can see that majority of a test and val is considered of high quality, whereas for train, well, we have to unfortunately, we don't have the data of a high quality at this high proportion, but so it's up to the user to decide if they want to kind of like filter a data based on quality or not, or maybe there's still some value in the training on the lower resolution data, provided that you have more of it. So another part refers to structure completeness. So that's already what I alluded to. So we can look at you can find non-resolved. So kind of like it's a ligand number of unresolved heavy atoms. How many of unresolved heavy atoms are in the system? Not per individual ligand, but all of the individual ligands summed together. It's also about the pocket part. So this is really to provide a lot of information for a potential modeler and method developer to use this data tailored to their requirements. You can have a look in terms of this case, it's unresolved residues in a protein sequence. So that's comparing the sequence from a sequence to the number of resolved residues. And we can see that from majority of systems you have, most of them are fully resolved, but some of them have a decaying fraction of residues that are missing. And in this case, it also will include a couple of cases in val and test. That's because what most parts that are unresolved typically is the flexible parts that are not around the binding site. So we can look at the percentages for the ligands as well. So like ligands are mostly resolved. The test is not 100% here. This is not a bug. It's because of the fact that we allow some of the covalent ligands to also be in the test set if a high resolution, because we have some of the leaving groups, and therefore the number of atoms that are resolved end up being slightly smaller than the ligand that was added as an input because of a reaction that happens by covalent addition. Zooming through the pocket domain, so it's important also this is for diversity purposes. We want to know that our systems are diverse and belong to different eco-domains. So it's encyclopedia of cobalt domains. So there's protein families, there's like different kinase annotations. So this can be also queried. And we can also get some of the value counts in terms of how many different proteins there are in the HIM barrels, or protein kinases, or globin-like. So for structural biologists, that might be of big importance. Looking at some of the kinases, we can see CDK2 is most prevalent in the crystal structure, and that might indicate some of the importance of this system being studied for pharmaceutical development. Ligand properties are described based on the ligand molecular properties, so it can be molecular weight, it can be hydrogen bond donors, but also some of the things that we chose is Booleans, basically whether it is a known kinase inhibitor and similar. So we can also see how these are looking at the proper ones, focusing on the ones that are not artifacts, not ions, across the different train valve test splits. You can actually plot them and see how they would look for these different molecular properties. So we can see that they roughly cover similar distributions in terms of molecular weight, number of heavy atoms, or number of rings. So kind of like this is an important sanity check that your test set is actually representative somewhat of a train and valve sets for valid comparisons. Finally, looking at some of the categorical parts again, this is where you can get the descriptions. And we can similarly look at some of the distributions for these categorical descriptions of how these ligands are classified. And we can see that majority of them are Lipinski compliant, this rule of 5. But the general trend is somewhat similar between the different splits, and that's something that is important in order for your data to be represented. Then going into protein ligand specific part, we have annotations for the crystal contacts, we have annotation for binding affinity, and we have a number of interactions the ligand makes, and even specific interactions listed as somewhat encoded fingerprint. So you can see we included some of the binding affinity annotations, and while majority of it might be lacking it, there are still some in test, val, and in train that are true. So that allows also perform different studies that require that information. And you can see there's also a range of distribution for these values. We specifically chose to only include PKI or PKD values, and there's a good reason for that, but those are more comparable between different assays run in different labs, and therefore can be more readily collected together. And this is looking at those interactions that I mentioned before. So this describes which chain, which residue, and what kind of type of interactions the ligand is making in this particular system. So this is just showing one example, but you can see that it hashed up whether there's a hydrogen bond, whether it's a side chain, or whether if it's not a side chain, then it's a backbone, hydrophobic contacts, and similar. Pi stacking, what kind of stack type you get. So all of this information is available. There's also more, as we said, beyond the split. So it's actually you can go into more detail there. Like we provide this information accessible, so the notebook will contain it. But for the interest of time, we'll skip it for now, and If we have time, we can come back to it later. Are there any questions?
Nothing that hasn't been answered, I think.
Okay, fantastic. So questions have been answered already on Discord, but feel free to pour them in. We'll try to answer them either directly there or offline later. Like this is intended to be on the ongoing conversation. So now the similarity clusters is an important part of the annotations, and this is because that's what we use in order to split our data. This is how we find what systems are connected in terms of information leakage, whether we have similar pocket, whether we have similar protein description. So this is the pocket identity metric and different thresholds. There's also LDTT metrics, there's similarity metrics of different kinds. And there are two types of these descriptions. So there's actually a strong component that defines whether the similarity is bidirectional or a weak component. If a similarity is only that some of our similarity queries vary depending what's the query system, what's the target system. So this allows us also to know that information leaks both ways. And there's also like different communities that can be described or different components. So without going too much into graph theory, we'll just go further to show maybe, do we have time for the quick example?
We can come back to it.
We can probably come back to it.
Yeah.
So we have a couple of examples to show how we can use these clusters for identifying interesting subsets in our data, as well as how it can be used to sample diversity. There's also beyond Plindex, so more annotations that are available, but they didn't make it to our short list of 745. So it's for exactly that reason that it would be not too overwhelming. But those are available for those that want to go into more of intermediate files. Are actually available on our Google Cloud bucket, and they can be loaded with some of PLINDER tools. So you can access them and have a description there, and hopefully some of you will find it useful. Now, it's probably like, since we've described a lot about what is inside and kind of what are great annotations, we'll go to accessing the system files, but before that, we might have a few questions.
Yeah. I think one question was, how are missing ligand heavy atoms identified?
Missing ligand heavy atoms are identified by comparing them to, so every ligand that is included in the PDB structure has a ligand ID, which is part of a chemical component dictionary. And from that, we can link it to the SMILES, which is like basically what is supposed to be the ground truth of those atoms. And then we try to match the result structure, but the model structure from the source PDB. We try to match it up with the SMILES, and those atoms that are not matched up, or like some of them might be potentially missing. So we take the difference between the two. So it is basically the number of atoms we expect in SMILES minus the number of atoms that are resolved and modeled. And similar, of course, for the residues, but obviously for residues, it's more known. It's only 20 amino acids, so typically you know how many atoms are expected there. So we match them up quite easily, and it's easy to make count.
The other question was, okay, it's a bit of a misnomer in our notebook, is that when you write number of ligands, index.shape 0, does that mean each ligand forms only one system?
So as we've already referring to here, for the, I think in this case, when we looked at, our systems are, by definition of a system is that we take a PDB entry, we provide all the biological units, we identify the ligands, and then identify all the protein chains that are within 6 angstroms of that ligand. So it could be more than one protein chain. We also, if a ligand is in contact, and in this case it's 4 angstroms, with another ligand, we merge them together into a single system. So as a result, you end up having some cases when you have systems with multiple proteins. We also some systems with multiple ligand chains. So like you can see like there are 6,795 that have 5 ligands in one. In this case, this is not necessarily a proper ligand, how already Tom has said. So this would include some of the ions. It would include some of the artifacts as well. So you can also, using the annotations that we have, you can make your own counts using the proper annotation, or you can define your own. But in principle, you get many multi-ligand, multi-protein systems as well, just by some overlap. But majority of it is probably like still single protein, single ligand.
Is there a way to filter out the ligands that bind at the transmembrane region?
Well, if there's a way for the transmembrane region, it probably would be by going through domains. If you know a particular domain that is appears in membranes, and that domain that is transmembrane region domain, then it would be a way to query systems that have that particular domain of interest. Or if you have a similar protein, but you know that it's transmembrane and you have some binders, you can also use the similarity to query similar systems. So those would be the approaches to investigate. But if you have any particular more detailed questions, we can also happy to answer them offline.
I think that's all of the questions so far.
Excellent. So the notebook is available at your hands. So feel free to query it. Feel free to ask the questions later. But now we probably all are itching to learn how to access the system files. And for that, I would like to call upon my colleague, Yusuf, in order to help us to guide through this.
Thank you so much, Vladas, for that detailed walkthrough. And since we've seen how to, access the annotations of these systems, we've also seen how to, look at the similarities. It's time to actually look at how do we interact with the systems, right? How do we interact with the systems in a way that do not require us to, have to chase down, the physical location of, the files, on our computers. And for this, we've created this sort of interface called the PLINDER system, where you can just specify the system ID, and this will return a number of useful path that you can actually interact with. And if you run this cell that we have displayed, you will get an example where this is directly just sort of linking to the system CIF. And that system CIF is what we are loading here, showing where the ligand binds on the protein, as well as the proteins. And in addition to this, like I said earlier, this provides you access to all the files that is associated with this particular system. If you look at on the next cell, you could also access the ligands directly. for each of this is, our systems are multi-ligand in nature. You have these sort of maps to their different chains, as well. And in addition to this, you could also get things like smiles, like we were showing here, you could get those smiles easily displayed here. And, the same thing also applies to, receptors. All the files that we have here, we have receptor files, this is in different formats. We have the CIF, we have the PDB formats. All these, could be made available by just simply using this interface, PLINDER system. And, in addition to that, we have things like in cases where you have, binding site water, we also have those mapped out where, we have files that is specifically, dedicated to that, and we can just sort of load that. In addition to that as well, we have sequences because, oftentimes we want to use, sequence as, one of our inputs, to train our models or for other use cases. We also providing, access to the sequence faster. And I have to point out that this is, the secrets of the particular protein. So this would have, full, sort of residue that are, associated with this particular system. In this case, you might have some residues here that is not present in the holo structure because this was supposed to actually provide those full sequence residues. And, having, said this, this is just sort of talking about, what are the files? How can we load them? How can we access them? The other angle, to this is, in addition to having holo structures, we also have linked structures, to the holo structures. These are APO structures. These are, predictor structures. In this particular case, AlphaFold predictor structures. And from this interface, you could also immediately just, access those, linked structures. And you can access those in addition to, their similarity metrics. And what we're showing here is just, the similarity metrics that we're focusing on, right? For link structures in this particular case, we want to know how, similar structurally the APO structure or the predictor structure is to the holo structure. In addition to that, we also want to look at, how well are we able to transplant, the ligand from the holo structure to these, new APO predictor structures. And for some of this, you've seen some of these metrics earlier from, Xavier's presentation. It's exactly the same, but in this particular case, just think about this in terms of, transplanting the ligand to the APO structures. And, this, sort of provide the basis for, selecting, the, APO structures that we might want to use in addition to the holo structure. And here we're, providing, all this information so that users can actually have the, freedom to, play around with these metrics and choose, which APO or predictor structures best suits, their need. Right. And if we look at an example here, we're picking the first example here, which is the first, instance on this data frame. You can see that, these as, identical, similar, identical in terms of, sequence identity. We have 100% sequence identity. In fact, all of them do have that. But you can see, there's, when you look at, the LDDT, the actual LDDT and the backbone LDDT, it's not exactly, the same. You sure you have a little bit of change in terms of the actual structure. And these are some of the things that we want to capture, in, these data frame that we have here. And, one important thing to note here, a lot of these metrics you've seen before. I think the one that you've probably not seen, and you might be wondering what this actually means, is the sort score here. And this is, our way of sort of assigning quality to these APO and predictor structures. And in this particular instance, sort score means, resolution for the APO structure, and the PLDDT for the predicted structure, because we want to be able to sort of assign, you know, some quality to these structures. And, you know, if you scroll down, you know, a little bit, right, you know, now that we actually now see, you know, what this APO structures are, one of the other things that, you know, we might just want to skim over is, if you want to learn more, you know, there's, you know, some other information here if you want to, you know, learn how to actually create that data frame that we show above. We're limiting it to just 5 examples right now. If you want more, you can run this, you know, at a later time, but I think we're just going to skip over this in this presentation. And also, we also want to mention that this is also optional, but you can run this as well. You know, there are cases where, you know, we don't have examples of APO and predicted structures. You might want to do predict for some systems. We're just showing an example of how you could do that here with ESM fold, and we don't want to, you know, spend too much time on this. And, you know, the other thing here is, now that we've looked at, you know, APO structures and predicted structures, right? We know that for some, because these are, you know, by definition, not exactly the same structures, they're going to be different in some respects, right? In some cases, their residues might be different. In some cases, their actual, you know, number of residues might also be different, you know. And to use this in deep learning or any other, you know, method, you know, sometimes you need to actually match, you know, the APO structure with your OLO structure. And this is what we're trying to actually show here, you know, examples of how you can actually, you know, align and, you know, mask and also featureize, you know, some of your, you know, APO and, you know, predicted structures. And what we're showing here is just, you know, listing examples of, you know, cases where we're listing properties here. And some of the properties you see here are specifically related to masking. And if you go to the next block here, before, I think, yeah, so this is not exactly the masking part. Yeah, so I mean, specifically here, you know, we're just, you know, looking at the masking here. And a typical example of what I was talking about here is, you have a, you know, structure. In some cases, you only care about the, you know, C alpha structures. And we have provided, you know, a way for you to actually get a mask to filter that out. You know, in this particular example, we're showing how you can filter out backbone structure, backbone atoms, filter out C alpha, you know, atoms, you know, if that's what you care about. And also, you know, importantly, we're also showing, you know, in addition to, you know, this particular set of properties, you know, if you care about filtering your structure in, you know, some other ways that is not specifically, you know, about, you know, C alpha or backbone, we're also providing a way, you know, to actually do that, you know, provided, you know, you have you know the kind of atoms you care about, essentially, you know, in terms of the mask, right? So pretty much this is what we have about, you know, just sort of masking. And in this, you know, next block, what we have here is just sort of showing how you can access the actual structure of this predicted and APO structure, right? What I showed earlier was just showing the data frames, right? You know, showing data frames of what is linked to what and what is their similarity metrics. But what we're showing here is, you know, now that you know, seen those, you know, how do you actually get these structures, right? And it's not enough to get these structures. How do you, you know, transform those structures, the APO structures, to, you know, align and, you know, match your, you know, HOLO structures, right? To do that, we also have provided, you know, a method, you know, which we call the align common sequence. And in that method, if you scroll above a little bit. In that method, you have to where that function is. Yeah, right here, right. In this particular method, you have a luxury of, playing around with this, and specifying these parameters, how you want. In this particular example, we are, aligning, the APO structure as well as the OLO structure, and just sort of, making sure they match in terms of, which atom match, which atom. In this case, we're removing things that don't match, but that doesn't have to be the case. In your example, you might want to do something different. Right. So we have that. And what I want to actually show is if you scroll up to this structure that we had before, right? If you look at this structure, this is APO, which is the predicted structure is shown in red. And the OLO structure is in blue. But what you can see clearly here is that on the C and N termini of the predicted structure, you can see the wiggly structure just hanging off, right? And those are not present in the OLO structure, right? After, aligning, you can see, this is, we have methods to just sort of align them and, strip out those part of the structures that are not present in the OLO structure. So this is, one example of how you can, access the APO and predicted structures and make sure that they align and match the OLO structures. And, I think this sort of, marks the end of the masking, aligning, and matching. And, the next part of this would be the loader, and you're just talking about the loader. But before I go into that, I don't know if there are any questions yet.
Thanks, Yosef. So this is super helpful, and having this aligned obviously will help a lot of people to match up their predictions going, especially from APO to HOLO, and those are the relevant inference and endpoints. Just before we go to the data loader, I think there's also one section that may be worthwhile going a bit over more. I'm happy to kind of do that as well. So one of the things that Xavier was also explaining, and for the protein ligand predictions, is sometimes mapping ligands is not so trivial. And that's why we provide not only these elaborate maskings that Yusuf so helpfully explained, and how to map them between APO and HOLO, but it's also between ligand structures. And for this, our molecules and the ligand molecules, what we end up providing is the input ligand template, which is a two-dimensional aldehyd molecule generated from a canonical aldehyd smiles. We also provide random conformer for this smile so that you can use as an input in case your method requires an input conformer generated. Then we also provide the coordinates, of course, that is linked to it. So this is a tensor that easily can be queried. We provide similar information for the resolved ligand molecules and resolved ligand coordinates. And then we provide something that we call here templates to resolved atom order stacks. So this might be new to some people, but it is an important feature to use. So unlike protein sequence, the ligands don't have a canonical ordering, and multiple valid ligand representations in terms of how atoms appear in a sequence are valid because it's defined by the graph. So as you can see, if we define our structure from smiles, and this is, here we read directly the object that is part of a PLINDER system. So input templates, ligand templates, here we provide the numbering, and the numbering is exactly how it appears in smiles. If we make a conformer, the conformer numbering stays the same. However, in the experimentally resolved structure, there's no guarantee that the ordering is the same. And for that reason, we need to provide the array that provides the order, how these two, essentially your input conformer and your output ground truth structure, how these atom orders should be compared. Because obviously, if you take the two X, Y, Z matrices and you subtract them, you're not guaranteed to give the sensible result. So here we would take the input atom order stack, and then we can take the hollow atom order stack, and these would be the kind of well, distances that one should compare. I think because we have a little bit of time, I'll just kind of illustrate this with a bit more complicated example where we have symmetry in the ligand. This particular ligand has a rotational symmetry around this bond, and in these, depending on how you generate this, these two atoms, oxygen 7, oxygen 18, are indistinguishable just from the smiles. And then, but if you resolve the structure and you choose only one ordering, then the distance between these two atoms might be very different, and you might be calculating wrong losses. So when mapping to the conformer, one needs to have a careful look which of the ordering of atoms is more relevant. And that's why, while this is while this ligand is perfectly resolved, and while this ligand is, like in the previous case, like you had only one atom order, here but it has two atom orders, so therefore you are provided with two sets of stacks. There's also some symmetries that can arise due to not only the intrinsic symmetry in the ligand, but also sometimes there might be some ambiguity for partial resolved ligand. For example, here you see a very long terpene molecule and or terpene-like rather. So it's almost like a polymer, and this snippet that is resolved can actually match at least 3 different parts of original templates. So as a result, we also provided the 3 sets of stacks, how to map up particular coordinates between your prediction and the ground truth. Hopefully that makes sense, and feel free to experiment with this later. So are there any questions? And so there are no questions in Discord, but if you do have some questions, please do ask. We'll answer them not necessarily directly now, but we'll stay there later and we can continue discussing there. So now I think I can ask Yusuf again to take us through the data set and data loader functions.
Thank you so much, Vladas. Thanks for actually going back to that. I skipped it through that during my talk. Yeah, so I mean, I've now learned about the different aspects of the system and how to interact with PLINDER on a system level. We want to also take this time to introduce how might one interact with PLINDER as a data set. And with this, we have provided yet another interface called the PLINDER data set, which try to actually provide access to PLINDER, PLINDER systems as a group, and not just as a unit. And this interface has a lot of different parameters that you can use to sort of initialize it. And a lot of the ones we're also actually just showing here, you can pass these sort of parameters to the data sets. And I think this is probably updated, but the key thing here is that for this interface, you have a split parameter where you can use to specify some kind of split. In this particular case, maybe train. or a test set and or even validation set. And in addition to that, you could also provide things like filters, right? Sometimes you don't want to look at the whole data set. You only want to look at, for example, what we are showing here, you only want to look at systems, with only one protein chain, for example, right? We're also providing access to be able to actually load that, and for ligand, you could say you just want something that has ATP, if that's what you care about. And, these provide, the ability to, load the system in a way that users, want, giving them the freedom to choose what to include in the data set as they, load the data set. And, once you do this, typical, we're showing, an example here of what an item might actually look like in this particular case. And an item in this, example is just, PLINDER system. Right. And, all the properties that comes with it, you could access all the properties that comes with it. And the PLINDER system, as we defined earlier, could also have APO and OLU and all the things that sort of goes, with that. And, in addition to that, the data set also takes in a featurizer. Right. And one of the things, about, that is, this is a way you can actually turn your structure into some features that you can then feed into your, deep learning or machine learning model. And, that's, one of the other things. Right now, we provided, a default, featurizer, but that's by no means, set in stone. We encourage users to, come up with their own, featurizer, as, it's probably going to be the case, people care about, different ways of featurizing, these systems. And we want to provide people the access to be able to do whatever they want. The only requirement though is that, once you have to make, you have to make your featurizer in a way that it's compliant with, our own API. And we're providing an example of actually how to do that. If you scroll up, I don't know if I put that, I think that link was, yeah, so there's an example link here where you can click and just, see how that it's, done, right? And we also know that a lot of our, users also use, PyTorch, as their main tool for training models. And we are also, providing an example of how to actually make a touch loader, which, we've sort of implemented a simple example. And if you scroll down, I think I also have like a snippet. Okay. Okay. I don't think that was here, but you can actually look at, what we did here. It's nothing, fancy. It's just, wrapping, the data sets, around a touch, data loader. And you could immediately have access to that, and you could, loop through that, to train your model. The goal is to just sort of create, this, different batches of, features that you can then immediately just feed into your models. And I think that covers it for the loader. And at this point, I will also pause again to see if there are any questions before we go to the next section.
Yeah, and we also just posted a quick poll on like which other topics from this notebook maybe we could go in more detail if we have time at the end of the session, or also offline. So feel free to select that if we haven't covered a topic that you're specifically interested in. And of course, if you have any questions, you should. Maybe we give 2-3 minute break since it's been.
Yep.
Yeah.
Yeah, so during the break, feel free to query back the notebook and mark which question you would like to go in more detail. We'll come back there. We're also happy to continue answering your questions on Discord. So thank you for bringing them up. We'll be back in
5 minutes.
So in 5 minutes after the hour, so that is in 4 minutes from now.
Mhm. Okay, maybe let's continue. So I can go through now the evaluation part. So basically in PLINDER, we provide a couple of command line functions that make it easy for you to evaluate your predictions, and then also link them to the stratification, so the similarity scores that we were discussing before, to make the kinds of plots that we showed in the first session, right? Where you can compare the performance of your model to the similarity of each system to the training set. And so PLINDER eval, that's the command line tool, which is simply a wrapper around the open structure scorer as well as around postbusters. The postbusters is this other tool that gives you the physical plausibility of your predicted pose. And what PLINDER eval does is basically takes a prediction file, which is a CSV file containing just an ID. So each row is about a prediction. So you have to have an ID, which can be anything that you call your file name, right? The reference system ID, so this is the PLINDER system that you used, that is the ground truth. The receptor file, so that's the path to the predicted protein. If you leave this blank, then it assumes that you are doing rigid docking and it takes the system's receptor file. But fill this in for, with an apo structure if you're using that, or if you're using co-folding, for instance, then you have the predicted protein. The rank, this should be probably in the reverse order. So ligand file also, which is the path to the SDF file of the predicted ligand. And then for the prediction, you can assign a rank, a one indexed rank. So if you have multiple poses predicted. And basically, if you run this through PLINDER eval, it gives you a scores file that basically has all of these metrics that we discussed. So LDDT PLI, busy RMSD, LDDT LP, and postbuster stuff. So I have an example here of running it with a single system using directly the Python function. So that's PLINDER eval docking evaluate. Let's see if it's already run, then maybe I don't run it because it takes a bit. But basically just made like a quick function that runs VINA, given a ligand and a receptor and a pocket center. And this was run on this system using this predicted structure as the apo structure, so as the protein. And this for the pocket center right now, we just use the centroid of the apo structure. So this is something like, it's not a deep learning method. It's quite a simple thing just to show how evaluation works. But just to demonstrate, like once you have ligand files like this, what also PLINDER eval does is simply run this evaluate function on the receptor file and the generated pose. And if you have flexible as false, that means it's rigid docking. And if you don't set the postbusters, then it only gives you the rigid the ligand specific score. So that would be LDDT PLI, LDDT LP, and busy RMSD. So as you can see here, based on the very low LDDT PLI values, in this case, VINA like this without a proper pocket definition did not give very good poses. I mean, that's to be expected. This is kind of a dummy example. You can also just run the same thing again with flexible equals true. This means that you will also score the protein. So you will get the LDDT scores, the QS scores of the entire protein as well. And with postbusters true, you would also get the postbuster checks. So for example, here you would now have LDDT and backbone LDDT and per chain LDDT. This is all just for the entire protein, irrespective of the ligand. And then this is how the postbusters results would look. So we've made the whole evaluation suite sort of able to handle multiple protein chains, multiple ligand chains, etc. So that's why you see this AVE everywhere. That means average. So that's averaged across the ligand chains in the system. There's also WAVE, which is the weighted average, which is then weighted by the number of atoms in the ligand. So if you have multi ligand systems and one is an ion, then it wouldn't contribute so much. And similarly for postbusters, the reasons why they're like fractions like this is like it basically tells you how many ligands in your system have passed this postbuster check. If you focus on the single ligand, single protein use case, then all of these are only about that one ligand in that system. Just to, yeah, and this is just showing you how the so this is the predicted thing from Vina. It puts the ligand here in this way. And then this is the, this didn't work. Yes, nice. So this is the true system. So you can see that it did get the pocket right. So using the centroid as the pocket and then setting 20 angstrom around that, still kind of got the pocket right, but not. Okay, so that's just evaluation. And then we have PLINDER stratify and PLINDER plot. So stratify is basically this command that, given a split, calculates the similarity of each system to the training set. So this is not something that you would have to run because we have run this already on the split that we provide. So we have this for here I'm showing the validation set. So because what you would typically want is to use these kinds of plots to monitor how your model is performing on your validation set. So we provide this file set parquet file, which is the result of PLINDER stratify, which basically for each system in the validation set labels the maximum similarity of the system to a training system. So for instance, this one, there is a training system with which it has 50% shared PLI similarity and so on. And so this is for all of the similarity scores. And we also provide these things at the end, which is basically, does it have a novel pocket or a PLI? Does it have a novel ligand? Does it have a novel protein? And so on. So with these two files, so with the scores that you get from your model predictions and the stratification that you get from this well-set parquet, what you can easily do is then plot the performance of your model as a function of the distance to the training set. So this is with PLINDER plot, you would give the scores file as input and the val-set file as input, and you get plots like this, where you can see, if you only look, in this case, for example, if you only look at systems which have 50% or lower similarity to the training set, what is the success rate compared to all systems? So basically here, in this case, this is the plot from our paper. This was from retraining DiffDoc. And basically what we found for DiffDoc specifically is that it has a very strong dependence on having seen the PLI before. So if it has seen the PLI before, then it's performing very well. If it hasn't seen it, then it's not performing well. So you can basically use this to understand the inductive biases in your model and maybe also improve what you're giving as input, or like and augment certain areas of the data set to help improve how it's generalizing and what it's learning. Yes, so I think now would be a good time also, yeah, for questions, of course, and also to look into that poll. And yeah.
We do have a question about, for multi-protein and ligand systems, does that mean the ligand interacts with multiple proteins, or just that another protein is present in the PDB file?
Yes, so great question. It does mean that the ligand interacts with multiple proteins or is interacting with a ligand that interacts with a different chain. So this is actually, let me go back all the way up. Here it is. So the way that we define a system is for every PDB entry, we generate the bio unit. For each bio unit, we get all the ligands and then all of the protein chains within 6 angstrom of any of these ligands. And ligands within 4 angstrom of each other are merged. So if you have 2 protein chains in a system, either that means that one of the ligands is within 6 angstrom of both chains, or it means that there may be 2 ligands and one of them is interacting with one and the other with the other, and the ligands are close to each other. I hope this made it clear. Any other questions?
Well, there's another question possible to compare predictions with the closest structure in the database, not the exact context. So this is for the eval part.
Let me think. So in general, you are able to choose the reference because you can easily run the evaluate function and set your own prediction and set your own reference. But if you want to then also find the closest maybe I'm kind of misunderstanding.
If in cases there is an sample of references available, one could just run against multiple of those references and then choose the appropriate one. But this is rarely the case. We typically have only one ground truth. But yeah, in principle, ligands and proteins can be flexible, and if your reference is, for example, derived from MD simulation or something like that, one could imagine doing this, but this is not part of the example yet. So there are also a couple. So because we went over many concepts, it seems like there's a good distribution across different questions that would be useful to go over in more detail. But particularly the one that is with data sets and data loaders that is being asked. Another part is this evaluation stratification that has just been explained, and aligning and masking. So I guess people are itching to start using the data set for training. Maybe we should ask our colleagues, Yusuf, if you would like to elaborate more on the featureizing or data loaders.
Yeah, absolutely. So I think I can go into a little bit more detail about this. And like I said earlier, the goal here is to make this as flexible as possible. And, that starts with, giving people, the ability to write their own sort of, featureizer. And if you maybe click on that, what we are providing here is, the ability to provide, a structure, which in this particular case would be, the same structural abstraction that we described earlier that you could get, directly from the system, PLINDER system. So when you do PLINDER system dot holo structure or PLINDER system dot alternate structures, what you get out is the structure, right? So the idea is that you want to be able to actually take that structure and convert it to some kind of a tensor representation that you can fit into your deep learning model. And we've done that here. And what we did here was to just take a few examples of some features that we believe could be useful. And that includes things like the coordinates, unique coordinates if you're predicting structure. We took something like, the sequence itself, when you take this a protein sequence, how do you mask that, to the structure? Which residues are present, which residues are not? In addition to that, we also looked at, how do you create a full atom representation of your input sequence, right? So taking these few examples, this is by no means exhaustive because we know that users might, have some things that they want to, include, and they should be able to do that, but the overall goal is you have to, at least the way we had it set up is, you take in as an input the structure, and the output should be a dictionary. If you go to the end of this, it should be a dictionary of tensors, right? Which corresponds to the features that you are interested in, right? And once you do this, and we also provided an extra function to just sort of pad, this is standard, because protein chains will not have the same number of atoms. Sometimes you need to just pad because this tensor, you don't want to have a ragged tensor, and we're just providing that function at the end. I mean, so just sort of a utility at the end to just sort of pad your feature. And would expect, users to do this, once you create your features, so you can literally just copy this last bit, and you should work fine with your code. And overall, what you get out is just a dictionary of tensors, padded tensors, essentially. And that is what, automatically, because once you do this is, your featureizer. But then, in order to actually create a data loader, you also need a collate function, right? Because you have these features at, systems level, but you would be training at a batch level, right? So you need to be able to collate this into batches, right? So if you actually go to, I think it's in dataset. Right, awesome, yeah. So the idea here is that, this collate function is going to be passed into your dataset, PLINDER dataset. And what it does is it just sort of takes in your list of all your systems and make batches out of them based on, the features that you have, as well as, some of the other, keys that we are also surfacing here. And, specifically, these keys include things like systems IDs. In that batch, you want to provide their system IDs. We're also, surfacing the actual structure, right? Because for some reason, some people might still need the structure, even during training, or maybe they want to actually extract some, properties that is not necessarily part of their tensors, their feature tensors. So we're still including, the actual structure here as well. And including the alternate structure, the path, some people might want to go all the way to the actual, path, to want to load their structure with some other, tool. We're providing all that in addition to the, actual, features that we showed earlier, right? And again, this is by no means, set in stone. Someone might want to do this differently, but we just sort of provide this as a guide to show how you might be able to actually put all these things together. I don't know if that, covers, the questions or answer the questions a bit.
Thanks a lot, Yusuf. We have 5 more minutes in this session, so if you have further questions, now would be the time. I think there's, you want me to answer that one?
The cosmograph?
No, the prediction.
Yes.
Okay, so there is this question on, given a prediction, is PLINDER able to find the complex that corresponds to that prediction or the closest without any pre-label? And so, okay, so we currently provide all versus all similarities for the PDB, and you could think of doing a sequence search and then getting the systems which are in that cluster. One thing that is more like a feature request that is something that we would have to work a bit on, and maybe you can help as well, is to expose basically the FoldSeq and MMSeq databases of the PLINDER systems, which you can then maybe query against and get the scores out for your personal systems. But this is not something that's readily, I mean, there are ways to do this, but there isn't like a, let's say, a blessed way yet.
Okay, there's another one. Are the featureized values computed on each load access to the dataset? So are they computed, or are they computed once or cached for future access? So that it's probably a question to Yusuf.
So right now they are not cached for future access, but they could. And that's also where the flexibility comes in. But right now it's just being computed on the fly.
I could further add that the interest for us is actually to enable the users to continue to develop and expand on the features. It's not necessarily the intent of a Blender group is kind of like to provide all the different featureizers. It's more like providing the data set and ability to kind of use it and consume it in a good way. But there's multiple of featureizers that could be implemented, but it's not a plan to support these as feature requests at this moment. And yes, exactly. We hope we can find the ways of using our functionalities blended into your featureizers.
Yes.
Okay, anything else? All right, so since we only have 3 minutes left, I think we could just give you a quick thing on, so the break is half an hour this time, I think. Double check that. Yes, it's a half an hour break and we resume half an hour from now. I wouldn't want to say the time because of time zones.
At the top of the hour.
Yeah, the top of the hour, exactly.
So
And this will be in the other Zoom link, so the first one. So we'll all rejoin as a group. And I'd just like to repeat that this Discord will be open after this workshop, and we're really hoping that you will all maybe contribute to Blender as well. It's a community effort, and anything that any features or bugs that you find as well, please keep us engaged. Yes. All right, see you in half an hour.
Thank you, guys.
Thank you.